All right, powerful words. Thank you, Michael, for kicking us off here. So with that, I'm going to transition to our first speaker. Oh, thanks. Dr. Ed Cook. Ed is the Earl Bain Professor of Psychiatry and Director of the Center for Neurodevelopmental Disorders and Associate Head for Clinical Affairs at the University of Illinois Chicago Department of Psychiatry. You can see his distinguished bio here. He attended University of Texas Medical Branch at Galveston and completed both adult and child and adolescent training at the University of Chicago, and then moved over to University of Illinois in 2005. We're really excited to have him speak today on a very important topic, translating the complex genetics of autism spectrum disorder. So I'll kick it over to Dr. Cook, who's online, by the way.
Very much. Hear me okay?
Yeah.
Great. Really appreciated the introduction and pleased to be here. And I know it's Autism and a Genetics Symposium, but I'll bring the links together along the way. No commercial disclosures to report. We'll start with ASD genetic epidemiology. This is why so many scientists moved into autism genetics. Some of these numbers may have changed, but the point is risk to siblings is 20%, 20-fold higher than the population. After 2 are affected in a sibship, it's 1/3 to 1/2 recurrence risk for next children born. Obviously much higher, but what really drove everyone into the field that were not interested in autism or mental health in general was this finding of 90% identical twins. If one had it, the other one did. There's nowhere a much smaller increased risk to fraternal twins. And the genetic contribution estimates actually center more around 90%. This was published in Science in the 90s and drove a lot of our colleagues from more general genetics into the field of autism. And similar data drive geneticists into the study of schizophrenia as well. These are genetic etiology groups. This is also true in schizophrenia. De novo, meaning neither parent had the variant, but the person with the disorder does. Rare de novo events are predicted in genetically lethal disorders. Now, genetically lethal means a disorder that affects the chance of having children. This is true of both autism and schizophrenia. And the individual effect of these variants is often very large. I think one example people probably be most familiar with would be something like Down syndrome, where neither parent has an extra chromosome 21, but the child does. The aggregate of all of this is about 10% of ASD. That doesn't mean every patient that I see will have one that we know is the case. And I'll get into that a little bit later, but these are very, very high risk factors, often sufficient to cause the disorder. Then the inherited variants, the rare ones have large effect, and these are often examples would be recessive conditions. The common variants, this has been studied throughout psychiatry and autism as well as schizophrenia. There aren't as many hits in autism, partly because the data sets are larger in schizophrenia, and also because there's more rare de novo variation in autism. But now this is interesting contrast, because any of these individual common variants is not the same as some of the rare de novo variants, which is sufficient to cause the illness. But on the other hand, these common variants, their aggregate effect is large in ASD. It contributes to about half of ASD risk. and applies to most of the patients. So this is a genetic model of autism. It's interesting, it's remained the same, although when I first presented it, the 2 to 5 overlapping circles, which you could think of as, we thought of as individual variants, where if you had 5 of them, you would have ASD. Now each of those circles would be hundreds or thousands of common variants. And it's really a question of whether someone had enough of them to go over the threshold of the disorder. And then there are less complex cases, which are more the rare de novo events that I told you. Some are inherited. And I'm going to emphasize those quite a bit more because I think they're better clues to biology for the individual. They often are contributing to differences, for example, of additional medical challenges that someone might have in addition to autism. So we'll start with chromosomal disorders, insertions, deletions. This is an additional copy or deleted portion, not of a whole chromosome, but of a part of a chromosome. And I've listed here some of the most common deletions of a piece of chromosome. 7q11, Williams syndrome, 15q11, q13. You're going to hear a lot about that part of chromosome 15 today. So deletions there lead to Prader-Willi syndrome, Angelman syndrome, and duplications are going to be quite relevant to ASD, which is what I'll say in addition to autism. 16p11.2, 22q11, which you're going to hear about from me, but is actually a stronger risk factor for schizophrenia as well. And then 22q13 deletion. Of those common deletion or insertion regions, there's autism risk in many of them. The 7q11, not deletion, but duplication. Interestingly enough, the 16p11.2 deletion, ASD risk, I'm going to tell you the duplication also has schizophrenia risk. And the 22q11 deletion, we're going to talk about it's an overlapping schizophrenia and autism risk. So I mentioned chromosome 15. I'd spent quite a bit of time on. So this is sort of a diagram of chromosome 15, and there's not much happening on the small arm or P arm. The Q arm, the 1,1 to 1,3 region, which is actually the first subband of the first band, so it's really actually the first 3 subbands of the chromosome that has genes that matter. This would be the normal. If it's deleted on the chromosome that came from the mother, it leads to Angelman syndrome, missing this piece from the mother, severe or profound intellectual disability, general cognitive problems, and ASD risk, which is actually high in Angelman syndrome. Paternal deletions, Prader-Willi deletions, missing this from the father, exact same deletion region, but the origin matters. Mild ID, or maybe not intellectual disability, but other learning problems without general cognitive impairment, often related to severe obesity, and also obsessive-compulsive difficulties beyond the obesity issues. So those are deletions. Just think of this region as missing. I'm going to focus a lot on the duplications because of their relevance to autism spectrum disorder. This second grouping here is an increase of this region, a duplication within the chromosome. So the total number of chromosomes would be 46, would not have changed. Actually, technological advances were mentioned very appropriately earlier. These were actually very hard to detect with chromosome technology, and I'll show you an example of how modern genetic methods are enabling much of what we're now discovering. But this would be an insertion within the chromosome. I'm actually showing it on the maternal side because, as you'll see, those are the ones that greatly increase autism risk. There can also be deletions on the chromosome that came from the father. We think that has much less of these three possibilities, the two different kinds of deletions, the two different kinds of duplications. The duplication from the father has little or no effect in contrast. And then there's another grouping of an extra piece of a chromosome. And these are actually, they're easy to detect that there's an extra piece of a chromosome on the traditional sort of gray banded chromosome analysis. But you really do have to make sure that this Q11, Q13 material is there, because if not, it's actually not going to cause difficulty. So this, I just still see this patient regularly. About, I guess I saw her first in about 88, meaning 1988. And this is an example where she has this extra chromosome. This is a kind of banding where it's the centromere being, or staining where it's the centromere being stained, and she has, they're called pseudodicentric. They're basically the DNA of centromeres, but they're not acting as two centromeres. And this is actually from the larger chromosome prep showing the two normal chromosome 15s and this extra piece. And this is the first woman that I saw that really led me into an interest in this area. Severe intellectual disability, epilepsy, which has fortunately been relatively easy to control, repetitive behaviors, anxiety about lack of order. When she came to me, she had self-injury, mood lability, and she came to me on haloperidol, and had she remained on that amount of haloperidol for the last 30 plus years, she would likely have the side effect of tardive dyskinesia. But she had a good response to carbamazepine and an SSRI, removal of the self-injury and other behavioral problems, and she's actually doing quite well. This is an open label case study. This does seem to be more helpful for the disorder, and may be an example of, probably is an example of precision medicine, where knowing the genetic condition helps to define the treatment. Other things that you think that one might think would be more helpful, even for the epilepsy, aren't as helpful as carbamazepine. I like to point people to this parent group. One of the advantages of identifying this syndrome of duplications in this region is the families get together. I would talk with one of the parents, and they'll affectionately refer to other dupers that they've met at conferences. Knowing that there are similar problems is very helpful. Sharing at their meetings things that work or don't work is also very helpful. This is actually a picture. I don't show you the gray banded picture, but I'll just make the point that we identified this in a laboratory using microsatellite markers. And when we sent off for the gray banding, we got the report back that there wasn't a duplication, but this was one of the first advances, which is metaphase fish, where it's most obvious in this C figure that instead of one band here, there's actually two bands of this region. So even with metaphase fish, it wasn't as obvious on each of these figures. This led to interphase fish, which was easier to determine that there was an extra piece. Nowadays, we would send off, the first thing we'd send off is chromosome microarray. And chromosome microarray, this is not at all hard to detect. So as an example of moving forward with technology. This is how we determined that the duplication came from the mother and mattered less coming from the father. So Basically, these are markers along the chromosome in this region. You should have 2 different chromosome 15s, and you see that both of the grandparents had what you would see the usual markers. Now, the mother should of the grandchildren should only receive this set of markers or this set of markers from the father, in the same way that coming from the mother's mother, you see this 197, 131 over here, but it was easiest to see in this one where there's 3 distinct markers, each identifying a different piece of this region. And that's what led us to know that instead of getting either the 139 or the 147 from the father, she got both of them. And so now you have this father without autism, mother clearly without autism. Some might say, oh, well, she has it, it must not matter. But the key distinction here is she gets the duplication from her father, but as the mother, now her children are going to receive the duplication from their mother, and that leads to the difference in whether they're affected or not. This has since been replicated, and this is one of the most common rare variations in autism, and also in contrast to some of the others of equal frequency. This one is higher. If you have it, you're more likely to have ASD. Meaning about 90%, if you have that, will have ASD. This is the region that actually shows you the microarray results, so it shows you what's duplicated. There is a little bit of variation at the ends, but it's this middle region that makes the biggest difference. And one of the things that we looked at, we actually found it looking at this GABA beta 3 and related anxiety genes, partly because of our anxiety and being interested in this. It's probably this UB3A that's contributing the most to the disorder, because we know from Angelman syndrome that is the gene that drives it. This is the most important maternally, meaning coming from the mother, expressed gene in the region. And there's a lot of emphasis on understanding actually some early work on developing trials to reduce the activity of UB3A as a treatment in this disorder, similar to how there's already trials going on to increase UB3A in Angelman syndrome where there's a deletion. GABA beta 3 has actually, just from the latest analysis that I'll mention later, been confirmed as one of the autism risk genes based on some of the sequencing studies that I will show you. So I'm going to talk a little, I'm going to move to CNVs and SNVs, and I'm going to start with CNVs, or copy number variants. Now, the first thing to think about is those chromosome insertions, duplications or deletions I talked about before are copy number variants. Those are ones that were identified. You could see them on chromosome analysis, but with improved technology, we can see much smaller copy number variants. And I think of these as missing a paragraph or a page of a book. They're typically relevant when they delete or add genes, and they're usually de novo. If you notice the first case I showed you of the duplication 15, that family's unusual, but the parent of origin means that it was new in the affected grandchildren because they were the first that had the maternally inherited duplication. Okay. So these are de novo autosomal dominant. I had trouble with this whole concept because to me, autosomal dominant was something like Huntington, where you have this large pedigree and 50% of everyone has it. This is an apparent paradox because the parents do not have the variant, only the child. But the idea is that it only takes one single allele to have an effect. And also, if this child of these two unaffected parents without the variant did have offspring, they would have a 50% chance of transmitting that variation. So in that way, you could think of it as how a dominant disorder might start after it being de novo. And most of the things that we're going to talk about in terms of risk, the rare de novo events are going to be these de novo autosomal dominant events. And these de novo autosomal dominant disorders occur when the severity of the disorder is enough for less likelihood of having children. It accounts for a child to have a severe clinical condition with unaffected parents because the mutation is not present in either parent. This continues to be one of great interest. 16p11.2, you actually have to have more refined definition of it. We actually are starting to use more of a nomenclature in the physical nomenclature of base pairs that precisely define where this is. This was from our lab, where you can see the normal chromosome number here, the drop in this region, about 500,000 base pairs of DNA, too small to see under the microscope, as opposed to, let's say, the 15q11, q13, which was right at the threshold of detection with microscopic detection with those gray GM-sustained chromosomes. So once this technology developed, which is either done by SNP array or by chromosome microarray, more typically in the clinic, eventually, and even now, you can technically do this with complete genome sequencing, which is where we're headed. It's really more of a cost issue. So this defined a new syndrome. This is zooming in, very similar to the 15q11, q13 region. We become interested in the genes in the region, sequence them fairly quickly, and no specific gene in this region is sufficient to contribute to phenotype. And many of these copy number variants are probably related to multiple genes within the region, either being deleted in what I'm showing you or duplicated as well. So variable expression becomes very important to think about. And here, the 16p11.2 C and Vs are very important in terms of an overlap with schizophrenia. So deletions lead to autism spectrum disorder, intellectual disability, and obesity. The obesity actually being the most common thing that is present for everyone. Duplications can either lead to ASD or schizophrenia. Interestingly enough, there's a flip side where there's thinness and not obesity. Both are rarely seen in controls, meaning their prevalence is their penetrance. If you have the deletion or duplication, you have an effect of it, but the expression varies. So I mentioned, for example, deletions with ASD. Well, initially we were only studying with the sort of most advanced technology people with ASD, but since then it's become clear that the diagnosis with a deletion can range, or deletion or duplication, taking them all together, can lead to ASD or ID or schizophrenia, but also there are patients that only have language disorders. I would say at least having a speech and language disorder is sort of the minimum impact of this. ASD only about 25%, which shows if we're only looking for it in autism, we're going to see it only in autism. But when things get broadened out to everyone who's being tested, you learn this. One very interesting thing, and it's a good example of an interaction between the common genetic variants, also an interaction with some of the environment that comes related to SES and IQ. There's an average drop of IQ of 30 from the average of the IQs from the parents. And also, it's interesting in that you see this only 25% with ASD. Well, the test of the parents shows an interaction with where we're all on the autism spectrum. Some of us are on the opposite end of clinically diagnosed autism. And the parents that were sort of closer to the affected with autism part of the spectrum were more likely to have a child with ASD with this duplication. The parents that had a very high level of flexibility, very strong in social communication, were least likely to have a child with ASD. And this is a very important paper from Marino De Luca about 10 years ago showing that. And that's not just for 16p11.2 CNVs, it's just the most intensively studied in this way. This is showing that where the deletions, duplications matter. And the key here is, and I would have liked to have been able to show this green part is possibly subtracting. There are some of these variants sufficient to cause autism by themselves. Let's think of the duplication 15q11, q13. There are others that are sort of at a threshold, and it may take other genetic risk factors and non-genetic risk factors, and some of those could push over the threshold, some could lower below the threshold. And there are some, and this would be paternal duplication in 15q11, q13, where, if you see ASD, it's that these other factors are sufficient to put someone over the threshold. Single nucleotide variants. Both parents, for example, might have the protein supercalifragilisticexpialidocious, a de novo missense in one of the child's 2 copies. Let's say this F is replaced by an X. I don't want to try to pronounce that, but as you see, it would change that. Some of these changes, you could actually see changing letters in here that might not make it that hard to pronounce. Others would. And that's a really good example of why some missense mutations disrupt the protein considerably, and other missense mutations have a benign effect. And then there's de novo nonsense. In this case, instead of replacing the X, at that point of a nonsense protein truncating mutation, the protein is only made that far, and the rest of it is not made. These are almost always leading to a loss of function of the protein, particularly if they occur earlier in the gene. So we've learned a lot from single nucleotide variant studies. Again, technology enabled when we were able to do whole exome sequencing. The exome is all of the DNA coding for protein, and the emphasis has been on finding these de novo single nucleotide variants in trios of individuals with ASD and their parents. To have the proband with ASD and their parents allows us to know that a change is de novo. And what we've learned overall is there's a higher burden for loss of function de novo SNVs and smaller enrichment of de novo missense SNVs in brain-expressed genes intolerant of missense mutation. Probably not surprising, they're brain-expressed genes. And the intolerance of mutation means some genes will tolerate even loss of function, meaning you can get by with just one functioning copy instead of 2 functioning copies, so that the evidence comes from the genes that you lose one copy or one copy is dysfunctional. And we use a Nomad browser, which is very helpful for looking for which genes tolerate these sorts of changes. This is the last publication from our Autism Sequencing Consortium group, and shows the various data approaches coming in, and we're basically ranking with very high significance these 72 genes from, in a sense, and many times they have higher levels of significance because of more cases found, more or less. What's really interesting about the coloring here is you see these loss of function or protein truncating variants like the supercalia I showed you, and then you also see evidence for this gene SCN2A, which is a sodium channel gene coming from missense variants, as opposed to SHANK3 here, which has, again, about half or so coming from protein truncating variants, but then a large number coming from deletion of the region and things that were picked up and are picked up under CNV analysis. So this is 72 genes. This was published 3 years ago. The current analysis that has been presented, and we're working on writing this up with the final analysis, is this number is now about 257 genes at the same strict level of significance. And this is, I have to say, has a lot to do with autism has benefited, as has schizophrenia genetics, from incredible amounts of sharing among investigators, as well as participation by so many families. This is just to show, and you'll see this later, in a later talk on schizophrenia. This is showing this similar rare coding variants in 10 genes in schizophrenia from about the same time. And it's these genes above this line here, although what our experience from the autism genetics has been that as we get larger samples, the group in this area largely moves up into that area. And the thing that I mentioned is that there's overlap with these 10 genes. 2 of them overlap with ASD genes, this gene called TRIO, and GRIN2A. Now, I note that GRIN2B is also an ASD gene, and mention that GRIN2A and GRIN2B genes code for NMDA 2A and 2B receptors, and these obviously related to learning, the synaptic plasticity, and there's already been work in this area in terms of developing drug treatments targeting these receptors, and it may be that might have different impact if you were someone that had variation in these genes or not. So I think I mentioned about a 10% increase in these rare de novo variants. I want to sort of see how you get to their exonic de novo CNVs, 5%, loss of function de novo SNVs, 10%. This is not limited to the 72 or now 257 gene analysis. Rare autosomal complete loss of function, 3%. Anyway, that all adds up to 20%, and so some will tell you that, well, we can find something in 20%, but fortunately that we participated in a study, the Simon Simplex collection, in which unaffected siblings were found, and if you add all those things up, you see about 10% of the unaffecteds have that, so there's an increase of 10% that's coming from those. And in some cases, like those 72 or 257 now genes I showed you, we're getting a better sense of which of those variants matters. But this is the difference between doing a burden analysis, very important for thinking about research, versus an individual patient that I'm seeing where it's very important to note is this particular variant is that pathogenic for this patient. So I'm really interested in the genetics. I think there's obviously interesting points that pedigree I showed you, or the family that I showed you with the maternal duplication 15q11, q13. That mom wasn't going to have another child unless she knew she could test for the presence. I don't know what she would have done with the knowledge, but she wanted the knowledge, and then went on to have a child that did not have the duplication. So there's a whole area of genetics use. I'll mention a little bit about that later. But to me, it's about how, and I appreciated the introduction so much, because the real question is, how do we develop new medications, both ones that are more effective, because we really don't have anything for autism that treats the core symptoms of restricted and repetitive behaviors and social communication problems. So how do we develop them? And some of the things we do have that may treat, let's say, associated irritability or associated other psychiatric disorders to have reduced side effects. So Fragile X is an example where there's a long history of moving from gene to fruit fly to mouse to clinical trials. There have been a lot of improvements in trial design. So far, this hasn't led to a clinical trial that's been positive, although some of this, that doesn't mean we're still not on the right track there. SHANK3 and insulin growth factor is an example where there's current clinical trials involved. And I also note that Rett syndrome, which was in the basically psychiatric manual, is often found in girls with ASD, girls with ASD 3 to 5, particularly when you had to investigate a specific gene as opposed to doing a whole exome and CNV approach or complete genome sequencing, now has an FDA indication for treatment with an IGF analog trofinetide. And that's, I don't think there's enough discussion. It's true that we would like it to be more effective, so more work needs to go into the area, but to have an FDA indication for a treatment for a developmental disorder that overlaps quite a bit with ASD, I think is important. And I think it may not be as much as where we want to be headed, but it's important to have milestones of success along the way. No doubt 30 years ago, I remember people saying, well, you can't treat neurodevelopmental disorders. And so I think we moved from nihilism to actually a pathway to treatment development. So I mentioned 72 to 257 genes, and, in one way, the 2 to 5 genes we thought we were chasing 30 years ago seems, in some ways, well, then we would have focused on those 2 to 5. And now we've got so many, but the interesting thing is to think about how they cluster. And this has been fascinating. If there's this learning pathway, frankly, I was less aware of. In fact, some of this, these led to a Nobel Prize since I learned about the synapse. But this whole pathway, which I would think of as the learning synapse, and if you and this really came from CNVs, if you deleted any of the proteins in this pathway, you had a risk for ASD or intellectual disability, or in some cases, schizophrenia. And first of all, shows how fine-tuned this system has to be. And when I say missing, I mean missing one copy, so dosage significant. It's interesting, we found, I think maybe one of the first, this is also called PSD95, we found one of the first cases of this, or maybe it was just one of the set of genes of PSD95, and it is very similar to SHANK, where SHANK2 is now implicated in addition to SHANK3. So some of these have paralogs, very similar genes, and in some cases, filling in the gaps of a table had to do just really with seeing more patients and having more patients in research studies. This is an example I mentioned before that in many cases what we're looking for is associated disorders. I think this is another practical point of genetics, which has pulling out all of autism spectrum disorder, understanding a specific etiology. This is another one. POGZ is the name of the gene. The syndrome is called White-Sutton. And this is another one where there is a group of people that get together, both people with White-Sutton syndrome and their families, and share interests. In fact, this particular family has been very good about getting the word out about her daughter. She's a woman in her mid-30s with ASD. She lives on her own with support, works on her own with support, both from agencies as well as her family. She also has bipolar mood disorder and OCD, and a lot of people in the audience will know that's a challenge in terms of our current pharmacological treatments. And then this was about 2019 to 2020. She always had a lot of worries, and she had worries about constipation. And what was striking was the fecal incontinence was not a sort of what you see, I guess, much more commonly in children, but you'll see also in others. She didn't have the typical constipation, sort of leaking incontinence. It seemed to be full. And she did get initial GI workup. I think it was somewhat impacted by her tolerance of the procedure, and I think it was also impacted by it occurring in April of 2020. And she developed acute severe prolapse of an elongated colon requiring emergency surgery. Fortunately, it was fortunate that it was an elongated colon because it was easier to find healthy colon to not as a one-step procedure. And what's interesting is in the pediatric group, as many as 15% in this syndromal form of ASD have surgical emergencies that are similar. They're different in pediatrics, but I think one of the fascinating things is when we had gone the traditional route and requested chromosomes, the insurance rejected it. Actually, they rejected microarray, much less sequencing, which was what was required here. A lot of it had to do with age. Like, is it going to matter? And this is an example where there are a lot of people with White-Sutton syndrome who are older but unidentified because it's mostly being identified with new technology in younger patients. Having had the privilege to treat patients for so long, I can now see course, and this is an example of letting people know, well, wait a second, they're not just pediatric surgical emergency risk. And I'll just add that another one of these, because you're going to hear later again about 22 Q11 deletion, also known as velocardiofacial syndrome, is another syndrome that is an ASD schizophrenia overlap with medical consequences. The other one I'll mention in ASD is PTEN mutations, which also are a cancer risk variant. So when they're identified, more cancer screening is indicated. So some ethical implications. I mentioned at least a little bit about the complexity of genetic effects and the 16p11.2 deletion and duplication. Recognizing the variation could be so high. And I think sometimes we may give the impression that it's less certain. I'm sorry, we may give the impression that things are more certain or we know more than we do. I think it's important to understand this complexity. Subthreshold traits, let's see if I have a slide for this. Yes, I do. So this is an example, if there were, we used to say 5 genes, but let's say there are hundreds of genes contributing to, for example, restricted interests. Well, that genetic risk may lead to, may be a contributor to autism and so restricted interests that someone has trouble engaging in the social world. But it also could be thought of as the ability to focus intensely. And frankly, many scientific contributions have been made by people that have this trait. Anxiety, I think as psychiatrists who know that this also means, may mean you avoid certain risks. So this is the point that subthreshold ASD traits are often adaptive for individuals without ASD, and frankly, for people with ASD, they may be strengths for them as well. And finally, the number of common variants contributing to ASD or schizophrenia means that you're never going to, people think that you're somehow going to do eugenics and not have a risk for schizophrenia and ASD, but you couldn't do that. All these variants are there. Many of them were there for a reason. And this idea of pre-existing condition, I think, means we're all somewhat predisposed. And, you know, some have ASD and it may present some challenges. Some of them may have been overcome by some. And so more implications. Genetic etiology does not reduce the need for rehabilitation, education, and non-genetic treatments. And the idea is to empower those with ASD and their families. Common risk variants of ASD are shared partly by all. So I've seen parents shift from the refrigerator mother idea of ASD, and I'm glad I mentioned this to people, and if they're not my age, they don't really know what it is, but it was blaming mothers for ASD, similar to having blamed mothers for schizophrenia. But then people go, oh, it's my genes at fault. And the point is, we all have genetic risk. And in one context, that genetic risk may be for ASD, but it may also have benefit. Connection to medical complications and treatments. So I mentioned before, identifying patients that may have increased risk for medical complications. And I did mention all 3 of those. And then the other thing I mentioned was developing new treatments through understanding of pathophysiology. To me, that's what it's about. I'm not going to see all those treatments developed, but I have patients young enough that they will, I think, benefit from them. And there's already benefits, like I mentioned, with the trofinetide and Rett syndrome. And I want to thank people with autism and their families who participate in the studies. Just even for our group here in Chicago is a large number. Everyone who cares for themselves or others with ASD, everyone who advocates for services and research in ASD, too many mentors to mention, trainees who have contributed and of whom I'm so proud, other collaborators, NIH, I can't thank enough, the Shaw Foundation, Autism Speaks, Simons Foundation, the Academy of Child and Adolescent Psychiatry, and others for funding. I want to add all these things, particularly those here. I would add schizophrenia to ASD as a clinical head for psychiatry and relative. I can't say enough about the importance of moving forward and improving schizophrenia treatment. And I want to thank all of you for your attention. Thank you.
So thanks so much, Ed. Phenomenal talk and certainly thought-provoking. We'll take a couple minutes for questions and hope that you'll be able to join us for the Q&A. So I'll kick it off, and it's a two-part question. So one is, what would be a takeaway you think for someone who is a clinician and working with patients with either an autism spectrum disorder or schizophrenia that you would take away from your research? And then second of all, what are you excited about in terms of either technology or scientific developments over the next few years?
The first one is, I think for this group, is probably thinking about treating the comorbidity of ASD. And I would say so many patients, I don't know. That's a big one. As far as the genetics, I think to think about, we probably should be thinking more about testing than we do. I think that's a discussion to have with people. I think it's happening at the pediatric level more, but if we're treating adults, we need to think more. I didn't mention, I think I did, the patient that insurance disallowed, the father self-referred, although I refer patients to the SPARK study. And the SPARK study not only is a major contributor to current developments in this research, but also if they find something that's clinically relevant, they actually get back to the clinician and let them know. And either work with the clinician who may, like in my case, feel okay with the genetic counseling, or work with the clinician to also have their genetic counselor work with the family. And the disadvantage, you don't get a report back showing that it's negative, so you're wondering if you're hearing. It is research. It's not what I would recommend, but it's a relevant thing to do. And did I miss a question? Oh, what am I excited about? Yep. I'm excited about the part of using this information to develop new treatments. And I think we're getting these large sample sizes and just going to 257 genes. I mean, just like personally excited because now we have these in front of us and we're seeing how they link up with do the ones and genes that are also expressed in the heart as well as the brain. Are they the ones that are contributing to what we need to follow up for the heart? But really, at the end of the day, and it may even be treating the comorbidity. I think some of the things that I would like to treat the most is some of this severe comorbid OCD that's often leading to severe problems for the patients and their families.
Super, thank you. Take a look here around the audience and see if there are any questions. Dr. Strauss.
Do you think there's any contribution, Dr. Cook, of mitochondrial DNA variation?
That's a really good question. There's definitely interesting mitochondrial syndromes, and you remind me that I don't think I got an answer to whether our whole genome, I think will pick up mitochondrial DNA. We have to make sure it's not getting masked. I don't think the exomes have been picking up mitochondrial, but I'm not sure. But I really like that question because it's something I asked about, and I forgot whether I got an answer to it. So, excellent question.
All right. Well, thank you so much, Ed. We really appreciate your talk and certainly kicks us off here. So with that, we'll transition to our next speaker.